In this objective question, from the given option, they are insisting us to specify the correct option illustrating why the person with blood group AB is considered as universal recipient. For this, let me understand the blood grouping system determining factor. In this particular chart, you can see the A blood group is determined by the A antigen and B antibody. The B blood group person is determined by B antigen or the RBC cell surface, and the A antibody is also called as anti-A. In the blood group AB individual, we are observing the presence of two antigen over the RBC cell surface, which is nothing but A antigen and B antigen. There is no antibody in the AB blood group system, unlike the O blood group person. In the O blood group person, we are observing both A antibody and B antibody is present, that we can call as anti-A and anti-B. Whereas there is no existence of the A antigen and B antigen in the O blood group individual. With this specification, we are choosing option A is a correct illustration about universal recipient AB blood group individual. Why? Because the AB blood group person is having both A antigen and B antigen over the RBC cell surface, and there is no antibody in the sense there is no A antibody and B antibody, but other antibodies are existing in that particular individual. So we are choosing option A as a correct answer to secure 4 mark after ignoring option B, C and D. What are the points we told with this tabular column that we are giving as a recap to take the NEET notes for future reference? For the first time, AB blood group type in human were reported by Coron Landsteiner. Blood group were identified by Landsteiner in 1900 for A, B and O, and by D. Castello and Staney for AB. Gene 1, isoagglutinin, determines ABO blood type. 3 allele are present. These genes are nothing but capital IA allele and capital IB allele. These two are dominant, unlike recessive allele I0 or small I. A antigen and B antigen are the name given to the protein that IA and IB allele produces. Antigen A and B are present on the red blood cell RBC of a people with AB blood type. However, they are no antibody present in the plasma for either antigen. With this specification, we choose an option A as a correct answer to secure 4 mark.